plasma membrane is the boundary between fluids within and outside of a cell. It also helps give the cell structure and shape and controls passage of materials into and out of the cell.
The cell membrane is said to be a phospholipid bilayer. This means it is made up of two layers of phospholipid molecules. The heads of the phospholipids, like water, and are hydrophilic, and thus face towards the fluid-filled outside of the cell and the fluid-filled cytoplasm inside the cell. The tails of the phospholipids do not like water and are hydrophobic, and thus face away from any fluids, burying themselves in the center of the phospholipid bilayer.
Aside from the phospholipids, protein molecules are either partially or wholly embedded in the cell membrane. Integral proteins are wholly embedded, meaning they span the entire width of the cell membrane. Peripheral proteins do not go across the entire membrane. They are said to be partially embedded. Some proteins in the cell membrane, called channel proteins or carrier proteins, help move molecules across the cell membrane.
Some of the proteins and lipids in the cell membrane may have carbohydrate chains attached to them. These are called glycoproteins and glycolipids. The carbohydrate chains that extend from glycolipids and glycoproteins in our cells differ from person to person. This is important as it allows our cells to recognize each other. For example, when a cell from outside our body enters our body, the carbohydrate chains on the glycolipids and glycoproteins will be different from those of our cells. The cells of our immune system may recognize this difference and react in an appropriate manner to keep us safe and healthy.
The membrane is a fluid like oil, and its components slowly drift past one another. The degree of fluidity is affected by the degree of saturation of fatty acid tails and the presence of cholesterol. The proteins in the cell membrane are scattered throughout the membrane in a mosaic pattern, which is why it is called the fluid mosaic model.
The cell membrane is said to be selectively permeable because it allows passage of certain materials into the cell but not others. The manner in which a material may move in or out of the cell depends on the nature of the material and the levels of concentration of the material inside or outside of the cell.
Diffusion is the movement of materials from an area of high concentration to an area of lower concentration due to random molecular movement. This is called movement down a concentration gradient. Molecules that can dissolve in the phospholipid bilayer and molecules that do not carry a charge can diffuse through the cell membrane.
Osmosis is the diffusion of water across a membrane. Like other molecules, water will move from an area of high concentration to an area of low concentration. The more solute there is in a solution, the lower the concentration of water in that solution.
The speed at which molecules will diffuse across a cell membrane depends on 4 factors. The first is the concentration gradient. If the concentration gradient is large, then diffusion will occur faster. Secondly, smaller molecules tend to diffuse faster than larger molecules. Thirdly, if the molecule is hydrophobic and so can dissolve into the lipid bilayer, it'll diffuse more quickly than a hydrophilic molecule that cannot. Lastly, like chemical reactions, diffusion tends to happen faster at higher temperatures.
Facilitated transport is another method materials use to cross the cell membrane. Facilitated transport is like diffusion in that it is the movement of materials from an area of high concentration to an area of low concentration. However, it is different from diffusion because facilitated transport uses a carrier protein to get the materials across the cell membrane. Channel proteins form a pore in the membrane for small charged molecules. Larger hydrophilic molecules like amino acids and glucose can't dissolve in the phospholipid bilayer and rely on carrier proteins to enter or leave the cell. Carrier proteins are specific, meaning each can combine with only a certain type of molecule because they change shape to allow the molecule to pass into the cell. Also, like diffusion, facilitated transport is said to be passive because molecules are moving with the concentration gradient and thus no energy is expended by the cell.
In some cases, molecules need to do the opposite of diffusion. They need to move against a concentration gradient. That's from an area of low concentration to an area of higher concentration. This is called active transport. Since active transport is going against the concentration gradient, it requires two unique circumstances not found in diffusion. Active transport requires a carrier protein to get the molecule across the cell membrane, and the use of energy to overcome the concentration gradient.
Large molecules that are unable to pass through the lipid bilayer of the plasma membrane leave the cell instead by a process known as exocytosis. After production in a system of membranes called endoplasmic reticulum, the molecules are packaged in a small sac of membrane called a vesicle. The vesicle moves to a sac of membrane called the Golgi apparatus. Their membranes merge and the vesicle releases its contents for modification. As the molecule leaves the Golgi apparatus, they are again packaged in a vesicle, which moves to the plasma membrane. As the membranes merge again, the contents leave the cell without actually crossing the plasma membrane. An example in our body of the process of exocytosis is when the cell of a pancreas releases insulin to the bloodstream. Endocytosis is a process where cells take in a substance by formation of a vesicle.
Phagocytosis and pinocytosis are two types of endocytosis. Phagocytosis happens when phagocytic cells engulf cells or other large molecules, which they then digest. Certain white blood cells use this mechanism to rid the body of bacteria and damaged body cells. Pinocytosis is when the material is taken in a liquid or solid particle smaller than the one taken in by phagocytosis. Due to the differences in the materials they take in, phagocytosis is often referred to as cell eating, while pinocytosis is often referred to as cell drinking.
Cells communicate by means of chemical signals.